Hello, everyone. New background again today. So, we've got the very first part of our Unit 4, which is Genetics and Heredity for my biology class. And the first session of this is about Gregor Mendel and his experiments on heredity. He bred pea plants, and he bred them over a long period of time, and he bred a lot of pea plants. He was able to do this because he had a greenhouse where he could grow them year round, so he didn't have to worry about winter or the weather, and they mature pretty fast in a matter of weeks instead of months. Right, so it doesn't take very long for a generation of them to grow up and be able to have offspring of their own. In 1866, he published his findings. That's not as always, I don't require you to memorize those dates. But remember, we're talking mid 80s here. This is right after the American Civil War. Those of you who are also studying American history at this point in time. So what he did is he took pea plants, which might have different colored flowers, might have different colored seed pods. They might have different textures of the pea itself. The pea might be smooth or it might be bumpy. And he bred them with one another. And he did this until he was able to start establishing a pattern on how they actually passed down those traits from parent to offspring. Right? So what he did was his primary study was on the color of the seed pod, and he would take green seed podded pea plants and breed them with yellow seed podded pea plants. And you can see that on this diagram here. The first generation, both offspring were yellow. So all the offspring in the first generation are yellow here. Now, when he took that generation and bred them together to make a new generation of offspring, this third generation in the diagram shown below, some of them, about 25%, were green, even though both parents were yellow. So he thought that there must be some hidden way for that green trait to be passed down, even if the plant didn't look green. And this led him to talking about alleles. An allele is just a different copy of a chromosome. So you generally have two, right? Because we have diploid cells, so we have two copies of a chromosome, where one chromosome looks a specific way and another chromosome is very, very similar, but it has a mutation on it. And that mutation causes a change in the way something looks or acts. It's not a silent mutation, right? So as these pea plants were continually bred, he realized that the seed traits are tied to differing alleles. All right, this diagram here shows the texture of the pea instead of the color of the pea pod. So we could see that he had bred a pea plant with a smooth pea and a rough or a bumpy or a wrinkled pea. And he eventually went on to say that the one with smooth here, that is homozygous for the smooth allele, he represented it with two capital letters, SS in this case. The homozygous for the bumpy P, he represented with lowercase SS. So the uppercase and lowercase here matter. Make sure when you're doing work for me that you pay attention to that because it's going to be important in this unit. And when they form gametes, they are going to form a capital S gamete from the homozygous capital S and a lowercase s from the homozygous lowercase s. When he bred those together, though, they would mix and they would be capital S, lowercase s for every offspring. But because the capital S is the dominant one, it's going to look like that. So the offspring of that first generation is all S. Now, capital S, lowercase s, and it's going to look smooth. So if we take this information and we go back to the seed pod color, right, we can see that the yellow ones are dominant because he has represented them with a capital Y, and the green ones are recessive because they are lowercase y. All right, recessive just means they don't show if there's a different version that is dominant. So that first generation, where it's a homozygous yellow and a homozygous green, when they are bred together, they make a yellow generation that are all heterozygous. Hetero means different, homo means the same, right? So heterozygous, different zygotes. So they've got a capital Y and a lowercase y for everybody. But when he bred those down again, some of them would be lowercase y, homozygous green, and that green would appear. Some of them would be capital Y, homozygous yellow, and the majority of them, about 50%, are going to have capital Y, lowercase y. So they're heterozygous yellow. All right. I know this is kind of confusing. We're going to take our time with this. We are going to talk about how to figure out what these look like. All right. But for now, remember that alleles are different versions of chromosomes where the specific gene that codes for something has been mutated to look different. And because it looks different, it acts different. Because form makes function in biology. Right? Homozygous has two of the same alleles. It doesn't matter if it's dominant or recessive. And heterozygous is two different alleles. Right? So when he did his study on the flowers, he did the same thing again. And the third generation looked like this, right? The phenotype is what it looks like. The genotype is its genes. All right. So phenotype on the side with the colors, and genotype with the alleles. Right. And this is a chart that just shows the ratios or the allele frequency for each type. All right. So the ratio of 3-to-1 for the colors, because 3 of the 4 are purple and 1 is white. So 3-to-4. 3-to-1, 3 plus 1 makes 4. All right. Genotype, the ratio is 1-to-2-to-1. I know my face is covering up one of the ones, but you can count. All right. So the homozygous purple, capital P, is the genotype for dominant purple allele, right? So there's one of those. Heterozygous, capital P, lowercase p, still looks purple, right? So the phenotype hasn't changed. The genotype has. There are two of those, which is about 50%. If you only have 4, it's exactly. But if you're doing a large-scale breeding, you might have some differences here. That's why I'm saying about, even though the numbers on the screen are exact. Right? So the last one here, the homozygous for the recessive, and we're going to talk more about dominant and recessive, don't worry. Lowercase p. The genotype is homozygous, and the phenotype is white. It looks different because there's no capital P present. It's all lowercase. Because it's homozygous and it looks like this, we can say that, yeah, that's got a different phenotype than the others. All right. So Gregor Mendel, actually kicked off a large portion of study of heredity, how genes are passed down, how traits are passed down from parent to offspring. His studies, which were on the average simpler, started a inquiry into how all traits are passed down through genetic inheritance. All right. We've already talked about how meiosis takes those genes from an adult, right, and makes germ cells, right? That crossing over is important. It changes the allele frequency, which alleles appear on which chromosomes that get inherited down to the zygote, the offspring of in at least humans, an egg and a sperm. In flowers, it's pollen and different parts of the plant, all right? So that whole thing kicks off a large study, and it happened way back in 1860s. He published in 1866. Right? As always, if you've got any questions, reach out to me. Have a good afternoon. or morning, I guess, if you're watching this in the